data from American Energy Information Agency shows that today oil represents 34% of the total energy supply. The same source estimates that world demand for energy will increase 45% by 2030. And the most optimistic projections suggest that renewable energy sources will provide only 10% of these requirements. Until there, we won't be able to establish a completely different matrix of energy. We will still need oil. But that may not be enough, because current technologies recover from 1/3 to 1/2 of the oil contained in a reservoir. To increase oil recovery has been a goal for every research institute dedicated to oil industry in the world. Unfortunately, solutions provided by engineering departments have accomplished the goal only until a certain point, after which the cost is too high to justify production. And despite that, little attention has been paid to biological solutions, while in many other industries such as health and food, biotech has provided dramatic increase in yield at low cost. In the energy industry, research has been limited to biofuels. The limitation of traditional microbiological techniques to grow and identify bacteria and fungi has led to an underestimation of the role of those microorganisms and biogeochemical processes associated with oil exploration and production. We didn't know exactly the diversity and abundance of microorganisms there, so we couldn't know exactly what they are capable of. Nowadays, we can follow an event like deep sea bacterial bloom that follow the eruption of gas and oil into deep water horizon event. We can witness the consumption of hydrocarbons in the water, formation of environmental anomalies, and change in the microbial community that end up altering the same bacterial community that started the process, in a feedback mechanism. Similar processes take place in a well during drilling and production. New microorganisms are inputted with injection water, and different local microorganisms are harvested by the production water and brought to the water column. Nobody knew, because until recently no one had really studied that. Nowadays we know that two oil fields in completely different parts of the globe can have a completely different microbiota. Actually, two oil fields in the same basin can have completely different microbiota. In fact, two wells in the same oil field can have a completely different biota. The scientific community has identified microorganisms that remove paraffin deposits, metabolize mobilized oil, reduce oil viscosity, dissolve carbonaceous minerals or deposits, alter wettability, emulsify oil, lower interfacial tension, plug water channels, and reduce permeability. However, this knowledge is limited, fragmented, and even though the potential usage of microbes to enhance oil recovery is clear, there is still no clear understanding of which and how to use microbial processes. We are convinced that the first approach has to be the microorganism identification in oil field rocks, sediments, and water. Our laboratory has accumulated expertise in identification of gene-environment relationships and its application in biotechnological processes over the last 15 years, and we are now building infrastructure to address the issues of microbial enhanced oil recovery, starting by the implementation of new generation sequencing platform associated to bioinformatical tools to study the metagenomes of oil field environmental samples. This way, in a near future, we can study the relative abundance and diversity of genes involved in specific MEOR process, occurrence of processes not yet encountered in ecosystem, and novel functions in microorganisms not previously known. And when the demand comes, we will be ready to provide the innovative solutions that oil industry will need.